Hey guys, thanks for watching ManusScience. Today I want to go over AP Biology's Unit 1. Unit 1 is going to be over the macromolecules and water. Water is going to be a polar molecule. It's going to have a negative charge on the oxygen side and a positive charge on the hydrogen side. The hydrogen bonds that exist between the molecules of water are going to lead to properties such as cohesion and also adhesion, where you have water sticking to other molecules. The hydrogen bonds are also going to lead to this high surface tension where there's a certain amount of pressure needed to break the surface of water. This is also going to play a role in capillary action where water, you'll notice, will go up these thinner tubes, and also evapotranspiration, the movement of water up the xylem of a plant. This is also going to play a role, adhesion mainly, whenever you have a glass graduated cylinder and you have the water sticking to the side, you'll see that the water sticks to the glass surface of the graduated cylinder. Excuse me. Water is also going to be less dense when frozen, which is also pretty obvious, but it's also going to play a major role in aquatic ecosystems. So the organisms below don't get trapped and smushed ultimately. It's also going to be the universal solvent where it's going to dissolve most molecules. It's also going to have a high specific heat due to cohesion. It's going to resist rapid temperature changes. It's going to play a role in homeostasis. The high heat of vaporization is going to be where it takes a lot of heat to evaporate water, and that's going to be due to the breaking and reforming of the hydrogen bonds. This is going to play a role in evaporative cooling, where whenever the sweat evaporates off of our face or our body, it actually helps cool us. A role in panting in other organisms, and it's also going to help moderate temperatures of cities. The ocean will absorb a lot of that heat without actually changing too much of its own temperature. But water mainly plays a role when we talk about longer polymers that are formed. Dehydration reaction, you'll see here, also known as synthesis reaction. Whenever we want to link together individual monomers to form a larger molecule, we're going to have to remove a molecule of water. For hydrolysis, that's going to be the digestion or the breaking apart of molecules. This plays a role in macromolecules as well as DNA. Now, the first macromolecule we talk about are carbohydrates. That's going to be carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. Glucose is the main monomer that we talk about. And you'll notice that there's no nitrogen or phosphorus in this molecule. Energy is going to be a major role of carbohydrates. Glycogen in the liver cells of animals and starch in plants. That's going to play a major role in energy and storage. And also the structure, the cellulose that's found in plants and the chitin that's found in the exoskeleton of bugs, insects, and fungi. Peptidoglycan in bacteria as well. And to form these polymers, we're going to have dehydration reactions to form the glycosidic linkages, and it's going to be broken down via hydrolysis, like we just talked about with water. Lipids is going to be another macromolecule that we talk about. You'll notice that the unsaturated fats, they're going to be liquid at room temperature. They're going to have this carbon-carbon double bond, which is going to play a role in plants. This little kink here is going to provide extra space and prevents the cells from actually getting too close together and freezing, and it's going to help maintain that fluidity. Saturated fats, on the other hand, they're going to be solid at room temperature. They're going to be packed very closely, and they're also only going to have single bonds. So every carbon has a single hydrogen bond attached to it. Excuse me. Phospholipids are the main lipids that we refer to in biology. That's going to make up the cell membrane of all organisms. You'll see that the structure of them is going to be this fatty acid tail, and it's going to have a phosphate group in the head area, and that's going to create this hydrophobic and hydrophilic area of the cell membrane and allow for transport in and out. Proteins are a major macromolecule that we refer to in biology. The proteins are going to have the elements carbon, hydrogen, oxygen. Nitrogen will be a dead giveaway that you're looking at a protein, and also phosphorus. And the amino acids are going to be the monomers of proteins. They're the building blocks of proteins. Essentially, proteins are divided into 4 sublevels: the primary, secondary, tertiary, quaternary. The amino acid sequence is going to make up the primary level. The hydrogen bonds that exist in the alpha helix and the beta plated sheets, that's going to play a role in the secondary level. The third level is going to be the 3D shape of the molecule, and the quaternary structure of proteins only applies to certain proteins, but that's going to be where there's multiple subunits that are basically come together to form that particular protein. And any kind of mutation in any one of those levels, the primary through quaternary, that may denature the protein. Proteins are going to have many uses for transport, enzyme structure, recognition, hormones, but enzymes are one of the major ones that we refer to. They're going to be biological catalysts that increase the rate of a chemical reaction. And keep in mind that they're not altered, they're not changed by the reaction itself. They don't cause the reaction to occur. And these enzymes are going to have optimal conditions. And they may be denatured or broken apart or become useless if you change the pH, the temperature, the concentration of these particular enzymes. They won't work anymore. Now, enzymes are going to basically have this active site. This active site is going to be where the substrate will bind to the enzyme. And if this active site is altered in any shape or form, the enzyme will be useless. The active site is basically going to be broken down into competitive and non-competitive regulation. This is going to be where either the molecule is directly binding to the active site and preventing the substrate from getting in, or it's going to bind allosterically somewhere else, and it's going to cause a change in shape of this active site, and the enzyme will be useless. Some common enzymes that we refer to is lactase or DNA polymerase, RNA polymerase. We also have ligase, helicases, proteases, etc. Excuse me. And these reactions are basically going to be broken down whenever we talk about enzymes into catabolic and anabolic reactions. Catabolic, I always like to think of breaking down or cutting down a tree. There's going to be certain criteria that are going to be common with catabolic reactions, such as the releasing of heat. It's also going to have a negative change in energy and Gibbs free energy. And cellular respiration will be a good example for catabolic reactions. Anabolic reactions, on the other hand, they're going to be building up molecules. They're going to have a positive change in G. They're non-spontaneous. Photosynthesis would be a really good example for that type of reaction. Nucleic acids is the final macromolecule that we refer to, DNA being the first one that we mention at this point. It's going to be the genetic material. It's going to have the sugar deoxyribose. It's also going to have this sugar phosphate backbone. And the bases in between are going to be adenine, thymine, cytosine, guanine, with the cytosine, guanine having a triple bond and the adenine, thymine having a double bond. RNA is the other nucleic acid that we talked to. It's typically going to be an intermediary between DNA and proteins. Some of the examples would be mRNA, rRNA, and tRNA. They're also going to have that sugar phosphate backbone, just like DNA. They're also going to have certain bases in common with DNA, such as the adenine, cytosine, and guanine. But you'll notice that they also have a uracil. So they're going to have a slightly different base. And the sugar is going to be also different. That's going to be a ribose sugar. And these hydrogen bonds in between are still going to apply. So we still have a triple bond between the cytosine guanine, which is, again, a hydrogen bond, and the adenine thymine are just going to have a double bond. Whenever we want to make these molecules, they're going to be formed through dehydration reactions, like we mentioned in the water section, and they're also going to be broken down by the hydrolysis. And that will do it for our first unit. Thanks for watching.